All right. We continue this morning's program with Professor Shankar Balasubramanian's talk. He will be speaking on DNA. What is it telling us? The Q&A session will be moderated by Prof. Patrick Tan from the Agency for Science, Technology, and Research. Prof. Shankar, please.
Thank you. Well, it's a great pleasure to be here and having a lot of fun, especially meeting all the young, exciting scientists who are here. So I'm going to tell you about a story that started as a sort of an accident, an unintended consequence of some very basic blue sky science that I was doing with my collaborators at the beginning of my career. That we turned into, I guess you'd call a technology that could do something quite useful. So I'll tell you a little bit about that process of transitioning. And we ended up actually starting a biotech company called Solexa as a vehicle to drive the development of a commercial product that could enable things to happen. And so I'll tell you a little bit about that. And there was a transition from Solexa to a company called Illumina. And what we started actually drives all Illumina's sequencing systems today. And then I'll end by sort of coming back to why did we do this in the first place, and what was the vision, and some aspects of the impact that perhaps it's starting to have, particularly in clinical medicine. So we heard a bit about this from Tim Hunt this morning, DNA and Cambridge. And in fact, at the very start of my career in Cambridge, in many ways, I was searching for a problem. And a really good piece of advice one of my senior colleagues gave to me at a time when I was trying way too many different things, a head full of ideas that kept changing. And he said, look, you need to calm down and find a problem that's worth spending at least 10 years of your life on and focus. And it's okay to struggle to find that problem. And so the start of this story actually relates to part of that. Now, here's DNA, and it's now about 71 years before the critical papers that came out from Crick and Watson and Franklin and Wilkins that told us that the molecular mechanism by which genetic information is transferred and stored actually boils down to something incredibly elegant and simple at the molecular level. And that is the shape and hydrogen bonding recognition of C and G and T and A across one strand to the other strand in the double helix. And this information is stored in the middle of the double helix down this axis here. So it's kind of buried on the inside. And so DNA is a linear polymer in each strand. And what is key is the order and organization of these bases that we abbreviate to the four letters G, C, T, and A. And the Watson-Crick molecular recognition model explains how one strand of sequence information actually interacts with the other strand. And this can bring two strands together, but it's also quite critical for replication of DNA. Because the information in this strand can provide a template in order to synthesize the complementary information in another strand. And this happens every time DNA is replicated during cell division. So DNA is molecular information, and I've spent the 30 years of my research career so far studying three different aspects of DNA structure, chemistry, and how it conveys information. I'm going to tell you just about one of those today. So you all know about the Human Genome Project, which started in the 1990s and took about 10 years to interpret the organization of the human genome. Now, the genome is the sum total of information in one cell. And one copy of a human genome is roughly about 3 billion letters organized in 23 chromosomes. And it's the ordering of the letters that provides, the fundamental basis for interpreting the information. And this, of course, is organized into units called genes. There are now protein coding genes, which forms about 2% of the genome. And the remaining 98%, which is not protein coding, and it's sometimes referred to as non-coding, also turns out to be functionally important, whilst not coding for genes. So really, the drive of the Human Genome Project was to define what this structure looked like at base resolution. And this was, of course, fundamentally important to understand living systems, not just humans, but indeed other organisms, including small organisms and bacteria and viruses, including RNA viruses, and I'll say a little bit about that later on. Now, the aspects of the Human Genome Project also included understanding what it is that's the genetic basis of who we are, how we're different. And following on from that comes, what is the genetic basis for either causation of human diseases or predispositions to human diseases. Now, at the very start of my career, we were absolutely doing nothing to do with that. We were doing something completely different. And I'm going to introduce a few characters here. So my main collaborator was this chap, David Klenerman, who actually came last year, so some of you will know him. Now, my background is organic chemistry and enzyme chemistry. Now, David is very different. He's a physical chemist who had lots of interesting laser spectroscopy setups. And I met him because I got some comments back from a paper we submitted that demanded an experiment with a very specific laser that I didn't have. So I went round the department banging on doors to see if anyone had a laser. And then someone said, you should talk to this guy, Klenerman, who started the same time as you, because he's got lots of cool lasers and he might be able to help you. So I met David, and it was correct. He had an amazing array of lasers and optical tables and so forth. And we quickly did the experiment. The paper got accepted. And then we continued the conversation over a series of meetings, just exploring each other's interests. And we really spoke quite different languages. But we thought up an interesting project and wrote a grant that got funded. And the project was to see if we could watch a machine called a DNA polymerase. Now, these are amazing catalysts that catalyze the template-directed synthesis of DNA with very high accuracy. And what we propose to do is watch this process at the single molecule level, because single molecule fluorescence spectroscopy at room temperature was a pretty new thing at that time. And we thought by bringing together a completely new observation technique to look at a fundamentally important chemical reaction would be an interesting thing. And we wrote a grant, which I've still kept, actually, a copy of this grant. It was 2.5 pages long. And the objective was to do this experiment and see what we could learn. And it was funded. And the sad thing is it would probably not be funded today, because there wasn't anything useful even remotely implied in what we were trying to do. We were just trying to see what we could learn. Now, the setup for this involved building an optical single molecule detection setup. And we took synthetic DNA and we pinned it to a fused silica surface covalently. And we would bind the polymerase to this, and we color-coded the building block with a fluorescent dye so that as the machine would incorporate this building block here to extend this DNA strand being synthesized, templated by this template strand, we could watch it happen and learn things about it. So this is what we proposed, and this is what we did. Now, welcome to my office is something that I often say when I bring visitors to this place. So the chemistry department where I work is just behind this house here. And if you come out and walk down this street, there's this establishment. Now, there's a very, very famous pub in Cambridge, which if you go down the road here and turn left and go straight, eventually you end up at a place called the Eagle, where some interesting discussions happened 70-odd years ago. Well, this was our place, and David and I started our careers around the same time. And we didn't have offices back then. So actually, we had to go find somewhere if we wanted to just chat. And so we used to come here often at the end of the day, sometimes in the afternoon, actually, if I'm honest, especially if the weather was nice, there was a courtyard outside, and we'd sit and shoot the breeze, sometimes with our postdocs. Now, I want to introduce these players. Colin Barnes was in my lab, an organic chemist, and Mark Osborne was in David's lab, a physical chemist. They were key to the origins of this story. And in fact, Colin was employee number 1 at Solexa and played a key role in the commercial development of this system. So here we were thinking about the Human Genome Project because it was just down the road. And of course, as we heard from Tim this morning, Fred Sanger is a legend in Cambridge, and there's Watson and Crick, and actually Alex Todd, organic chemist, was a predecessor of mine. The last person before me in the chemistry department who actually took an interest in nucleic acids, and he helped solve some of the chemical features of DNA and RNA. So it was natural for us to step back and think about a bigger picture. And it was during a series of discussions that we saw a way of adapting what we were doing to do something potentially useful. Shown schematically here. So if you take a DNA template with a primer to be extended, and instead of just incorporating one color-coded thing, if you could somehow color-code all 4 building blocks, which would require some chemistry, and select or engineer a polymerase machine to tolerate any chemical changes you'd make to these natural building blocks. If you compete them all, because this Watson-Crick interaction is part of the basis for the accuracy of DNA synthesis directed by a template, by a polymerase. An A would go in opposite the T. And a T would go in opposite an A, and a C would go in opposite a G, and so forth. Now, our reasoning was that if we could control this process, so an A would go in and stop, nothing else happened. By imaging this surface, we would see a red dye that told us the identity of the building block that went in. And therefore, it would tell us the identity of the templating base. So we could read one base of information. Now, if we could somehow remove what was blocking the second step and go back to here and do round 2, we would see the T go in opposite the A. The reaction would stop. And we would detect this color at this site on the surface and decode a second base. And so cycle by cycle, we would progressively be reading this strand of DNA. Now, this is quite different to the approach that Fred Sanger developed. Now, the reason why this excited us is you could potentially array a vast number of DNA molecules on a surface. And by two-dimensional imaging, you could actually decode the sequence of all of these fragments in parallel. So rather like the electronics revolution that led to the microchip and smaller features, higher density, this sort of leads you down a pathway of increasing speed and capacity of reading DNA. So what we envisaged is if you take DNA from a source, such as human cells, fragment the DNA, we showed early on you could monodisperse these fragments covalently on a surface so that almost all the fragments were in optically resolvable regions of the surface without interference from other fragments. Then if you decode them in parallel, you could sequence at scale. So a very, very early calculation, this was quite crude in 1997, simply looking at how many fragments could we sequence in parallel? What are the kinetic rate constants that are likely per cycle? We arrived at a figure of about 1 billion bases. So we figured we could sequence 1 billion bases per experiment in a system that worked this way. Now, we paid a visit to the Wellcome Trust Sanger Institute to meet some of the early pioneers of the Human Genome Project. And one of our questions was, without meaning to be facetious, this is only one genome. We're all different. What about all the other genomes? To really understand the genetic basis of humans, you have to surely sequence at population scale. How are you going to do that? And it was clear there was no method at that time that had been conceived of that could scale. So we shared with them what we were thinking, and they gave us really strong encouragement to pursue it. This was quite important because we were at early stage. We had no business dabbling in genetics. We were physical chemists and organic chemists. And they helped us see the context of why this could be important. Now, slightly technical slide. This is what a polymerase does. This is the chemistry of how these enzymes work. Here's the template. This is the part of the DNA that's being extended. This oxygen is very important. That's this oxygen here. And this is the building block. The base can be G, C, T, or A. And there's a templating base that forms an interaction across here. Now, this oxygen attacks the first phosphorus of this triphosphate, and the diphosphate is expelled, and there are 2 magnesiums that are actually the catalysts for this reaction. So we had to hijack this process by somehow attaching a dye and somehow ensuring that it only goes one step at a time. We pursued this approach, attaching the dye through some chemical linker to the side of the base. This is a T base, the side of the T base that does not engage in this very specific Watson-Crick hydrogen bonding interaction. This part actually, if you will, it relates to the major groove of the DNA double helix. And we had to put what chemists would call a protecting group on this oxygen to mask it chemically. This has become known as a reversible terminator because Sanger chemistry is terminator chemistry. Now, shown here is sort of some specific chemistry for the first generation sequencing nucleotides. And we used a reaction invented very early in the last century by a German chemist called Hermann Staudinger. And this chemistry leads to cleavage here and cleavage here. And this works really well in water. And it doesn't cross-react with any other part of the DNA structure. Okay, so now we have a method for solid phase DNA sequencing when the DNA is attached to a surface. Now, there's one change to our initial plan, which was to sequence single molecules. Now, this was precipitated by a conversation I had with Sidney Brenner, who, of course, spent quite a bit of his time in Singapore, but I was summoned to his house in Ely because he'd taken an interest in what we were doing. And I remember a conversation in his kitchen. Just picture this. I wish I had a photograph. Sat around his kitchen table, and there are pots and pans and kettles and things lying around. And the essence of the conversation was that he persuaded me very forcefully that we should not sequence single molecules, but should amplify the molecules to get copies. And his argument was, if there's a mistake in the process at single molecule level, how will you know if it's a mistake or a mutation? Therefore, if you have lots of copies of that molecule, you can tolerate the odd stochastic error in the process and improve accuracy. So I was compelled by this. Actually, part of that story where Sidney was less happy was that he had his own idea for how we should amplify it, and we didn't like that, and he didn't like the fact that we rejected his method for doing it. But we adopted one that was developed by a French molecular biologist called Pascal Meyer, who had a company called Mantea. And this is, in essence, the process works by, if you flow DNA sample, again, into a flow cell that has a surface, we can ligate adapters to the ends, and we can graft complementary synthetic DNA adapters to the surface. And what happens is, by sequence recognition, you capture by hybridization the fragments, and then you can melt and essentially carry out a reaction that's rather like a PCR reaction, but it's restricted to 2 dimensions, where you melt, prime, extend, and go through cycles, and you end up with hundreds of copies that are located in the same place where the original single molecule fragment started. And now you can sequence. Now this also allowed us to use cameras that were a little bit cheaper, less heavy, and more practical to use, and build a more practical instrument. Now, 2005 was when this approach generated the first genome. It was a tiny genome of this bacteriophage. And shown in this expansion here are, this is real data, by the way. It's not a simulation, it's a real image. And each of these spots is a cluster or lots of copies that originated from a single fragment or a single clone of DNA. And it's being imaged by a CCD camera on an inverted microscope setup. And each of the colors tells us the identity of the building block that has been incorporated at that step. What happens next is we detach the fluorophores, everything goes blank, we wash everything out, and we do cycle 2. And the color may change depending on what the next base is that's being read. Imagine doing this on gazillions of different sequences simultaneously. Now, the way this method works is the fragments are generated, let's say randomly. It's not completely random, but we don't know what the fragments are and where they came from. But if you read, and early on we calculated that if you were able to read about 25 bases of a fragment without knowing where it came from, even if it had 1 or 2 base changes in it, we should be able to computationally realign most of them to the reference genome generated by the Human Genome Project. So this is computational. It's a process called alignment. Actually, what then happens is you oversample, you align fragments such as this, and you can read each base. Now, nowadays, systems can actually read hundreds of bases, and you can read from one end and then reprime and read from the other end. So it's much better than 25 cycles. Now, in 2006, Solexa, the company we started, released a commercial system that people actually paid money for to buy and use, and they were mainly genome sensors. This is the very first one. It was called a genome analyzer. And lo and behold, at the push of the button, it was able to sequence 1 billion bases of DNA, which coincidentally matched that little sort of calculation we did back in 1997. The dimensionalities were slightly different, but it was an accurate prediction. And at that time, 2006, this was roughly equivalent to the capacity of the whole world sequencing capacity as it was at the time we started the project in 1997. So it was actually quite a big leap. Now, in January 2007, an American company called Illumina acquired all this technology in the company, and I sort of transferred my scientific advising to Illumina for the next 10 years. And they optimized and improved the technology, and the more recent systems that look a bit like this were able to sequence several trillion bases of DNA. So this is another 1,000-fold improvement. So overall, from the time we started the project, the improvement in speed and capacity was actually greater than 1 million-fold. And the cost, well, today the cost of a genome is low hundreds of US dollars, and the Human Genome Project was of the order of hundreds of millions to low billions of dollars. So again, 1 million-fold change. So that's great. It's like buying a super fast car. You drive it fast one day, but then what? Now, the main driving force for us, and I think for everyone in the community who were in the early days of what's now called genomics, was how can we advance the case for improving human health, scientifically, clinically. Now, I'm going to give 2 or 3 examples of what's been demonstrated and where I think this may go in the future. I'm going to start with this population profiling. There are now well over 1 million human genomes that have been sequenced. Some of them are very targeted projects, such as this one, which is all about cancer. Some of them are actually part of an entrenched healthcare operation. Genomics England is actually part of the National Health Service in the UK. And it was created by former Prime Minister David Cameron, actually. He was a driver for this. And the aim here was to sequence hundreds of thousands of genomes of patients, mainly in the areas of cancer, rare diseases, and also infection. Other countries have the equivalent. The US has this project. The aim is to sequence 1 million genomes over 10 years. Now, one thing that all of these projects have, and I haven't mentioned the UK Biobank, which is 500,000 people who've given biological samples, is the data being generated is not just sequence data. It's healthcare data. In many cases, it's lifestyle data. And it's being accumulated over the lifetimes of individuals. So this is a multi-decade, transgenerational project. It's providing data, typically publicly, because it's made publicly available, to be mined to try and build understanding about how genetics and genomics may give us insights into disease and interaction with environment and so forth. So some specific examples. Here is a well-known mutation found in certain cancers. It's a mutation in a gene encoding this protein BRAF, which is a signaling protein. And when this specific mutation is made, it leads to an amino acid change in the protein that keeps the protein signaling on without control. And it's relatively common in skin cancers, melanomas. And melanoma patients who are diagnosed with this specific mutation respond very well to a drug that targets the gene product and switches it off. So this is a sort of simple but real example of how genetic information can direct very specific therapy and point out which cancers or which patients are more likely to be responsive to a given therapy. Now, projects such as this have at scale been looking at patterns of mutations, typically comparing the genome of the tumor with the genome of the patient pre-tumor, and even monitoring the tumor. What's emerging is, in some cases, patterns of genetic changes that are able to help physicians subclassify tumors. And in some cases, in clinics, the treatment regime is actually directed by the outcomes of such measurements. I want to say something about this because this is an area to look out for in the future. Tumor cells necrose, release their contents into the bloodstream. And part of the contents turn out to be fragments of DNA that no longer have cells associated with them. And it's been shown that by taking a blood draw and isolating these fragments of DNA and just sequencing all of them at great depth, you can pick up signs of cancer that originated from the tumor. Now, the more recent studies have shown that another feature of DNA called an epigenetic feature that can be determined by sequencing can not only indicate fragments that originated from tumor, but can also give you information about the tissue from which that fragment originated. So there is one example of a clinical trial being carried out in the NHS in the UK of 140,000 patients with no diagnosis. This is by a company called Grail, where this process is being carried out, and they're giving interim reports of its ability to detect up to 27 different tumors early from a relatively non-invasive process. So this is work in progress, and I think over the next 10 to 20 years, we'll see the sort of full extent to which such things can lead to benefit. Now, the other area where this is having an impact is rare diseases. Rare diseases collectively afflict 1 in 17 people on the planet. They have a high tendency to be genetic in origin, and when they manifest symptomatically, it's usually very, very early in life as a developmental disorder. And a series of studies called TRIO studies have shown by sequencing mum, dad, and child, you can find out genetic changes that are unique to the child. And 1/3 of the Genomics England work has been on rare diseases. And they have reported statistics of 25 to 30% of cases where there was just no diagnosis and no clue as to what's going on. They pinned down the genetic cause of the disease. And a fair proportion of those were metabolic disorders that could be treatable, for example, by nutritional supplements. And others are not yet treatable, but actually finding the genetic cause is the beginning of starting to understand mechanism and think about therapies. Now, the last area is actually infection, and I'll highlight this by relating to the pandemic that we all experienced very recently. So in Wuhan, the SARS-CoV-2 virus, which has an RNA genome, was sequenced using next-generation sequencing from a complex biological sample using a technique called metagenomics, where you sequence everything and you computationally piece together genomes. And this is how the SARS-CoV-2 virus was identified as a new strain. Then subsequently, during the pandemic, and there was a nice paper on this that came out in Nature, there was a database set up because patients around the world were having biological samples sequenced to identify the virus. And over 1 million patients around the world had the virus sequenced, and there was a real-time surveillance by sequencing of the emergence of new strains of the virus on a map of the world. Now, this has actually been quite useful for informing the sequence of the mRNA vaccines to be optimal and responsive to the new strains of virus. So I'm going to stop there and just leave you with these 3 messages. Thank you very much.
Thank you very much. It's on the far. Yeah.
Okay.
Oh, actually, you're supposed to sit here.
You want me to sit here?
Brief sit. Well, thank you very much, Sir Shankar. So it gives me great pleasure to start this moderation session. And I see when people come up, maybe I'll start the first question. So, we had David Klenerman come here last year, as you said, and I'll ask him the same thing that I'll ask you the same thing that I asked him. As institutions, how can we facilitate this sort of serendipitous interactions between different disciplines that, as you stated, was so integral to your that aha moment? And how do you nurture that and sustain that? Maybe I can ask your comments on that.
Well, you could start by having a pub that's nearby.
Okay, and NRF should take note of that.
Well, that isn't as ridiculous as it sounds, because we heard about the tea room at the Laboratory of Molecular Biology, and I know when EMBL was created, which was influenced by the LMB people, great attention was paid to the tea room and the cafeteria. And even today, in the new LMB, whenever I go to meet someone there, we chat in the tea room, and I'll bump into 2 or 3 other people, and we'll chat about our obsession and interests. And so I actually think it's difficult to force it and have productive results. You have to encourage it in a rather sort of subtle and relaxed way, because I think the relaxed encounters, chance encounters, can often lead to something a little bit off-piste that's interesting. And my own interaction with David Klenerman was, I needed a laser of a certain type. That was the driver. And then of course, we just kept talking.
Right.
So I think having an environment and also having a bit of space and time to do that goes a long way.
Thanks very much. Maybe the lady?
Hi, thank you. Thanks for the fantastic talk. My name is Kirsten. I'm at University College London. The field of genomics is currently getting really excited about nanopore or long-read sequencing. And I was wondering if maybe you could comment on that and give us your thoughts on where that might go in the future.
Long-read.
It's a good point. In fact, there are 2 long-read sequencing approaches. There's nanopore, and also a company called Pacific Biosciences has a different long-read technology. Now, the approach I've described gets you reads of 400-500 bases in length that you can realign. Now, there are certain types of information where you need much longer continuous pieces of sequence information. And so I think here techniques such as nanopore and Pacific Biosciences real-time sequencing is able to provide information that you can't get from this relatively short read sequencing. So I think these are hugely important and complementary methods that are being adopted, I think, more and more.
Thanks.
Great, thank you.
Lady?
Thank you, sir, for the insightful talk. I'm Himashi Sharma from India. So could you please enlighten us with your journey towards G quadruplexes structure. And my second question is that based on the information that we have about G quadruplexes structures, uh, will it be acceptable to assert G quadruplexes structures are the primary master regulator to regulate the gene expression?
G quadruplex.
Gosh,
Wow.
that's a whole new lecture.
Yes.
I'll give a brief answer. So G quadruplex is actually a four-stranded structure. And what really motivated me to work on this, we started again by accident. I went on a sabbatical to Bangalore, and I came back and found one of my PhD students had been doing completely different experiments to what I told him to do. And he'd met one of Aaron Klug's students in the bar at Christ College. And they did an experiment using phage display, and they selected a zinc finger protein that could bind to these funny G quadruplex structures in a test tube. This is how it started. It was my student hanging out in the bar. So, again.
Go to the bar also.
Now, fast forward to 27 years on. What really motivated me to work on this was the fact that friends and colleagues told me this was a useless waste of my time and a figment of my imagination. And why didn't I work on a decent problem? So I found that strongly motivating.
Yes.
And it's taken more than 25 years to have people make their own conclusions based on real data rather than their preconceived ideas of the world. So sort of a message I'd give to you is if anyone ever tells you not to do something, think of it as a strong motivator to do the opposite, because life isn't all about following conventions. And I think certainly discovery, you're not going to discover research because someone tells you what's going to happen.
Great. We have time maybe for two more questions because of the time, but Sir Shankar will be available during lunch to take other questions. Maybe the gentleman here, and then maybe one from that side. So maybe.
So my name is Daryl from the Karolinska Institute. So now we've had several generations of sequencing, so Sanger and GS, long read. So I'm wondering from your perspective, what do you think the future of sequencing is?
What remains to be.
Well, I think I'm not sure sequencing technology improvement is an exciting area for the future, because it's look, we're already around the sort of $100 to sequence a human genome at depth and pretty accurately. We might get from $100 to $50, but I don't think it's going to be transformational. I think two frontiers that are interesting. One is, of course, there are modifications to DNA and modifications to RNA, and we don't really understand what they're doing. So I think that's an interesting area to factor in to that research. The other part is really projects like Biobank, these large-scale projects where not just sequence data, but other data is being collected faithfully and made openly accessible, combined with new data analytics, possibly AI and machine learning. I'm not an expert. I think mining all that and trying to turn all that raw data and information into insights is, I think, perhaps the next challenge, at least in its intersection with human biology.
Great. Maybe one last question.
Thank you so much for sharing a fascinating story. I am Jude from Biologic Center, Thailand. I have a question. Genetics is known to impact the pathogenesis of the disease, but there are also another component like epigenetic changes that also contribute to the pathogenesis, like cancer as well. And I'm wondering how can we sequence these epigenetic changes?
Epigenetic changes.
Yeah.
Okay, well, that's, again, another lecture. So there are three things I've worked on, and this is the third. So there are now methods to do that. And in fact, exactly a year ago, I published a paper. Again, this is something that's been commercialized as a technology, of a method that you can use Sanger sequencing or next generation sequencing or anything, And you can get unambiguous information about GCTA, 5-methyl-C and 5-hydroxymethyl-C at read level. We call it 6-letter sequencing. And it doesn't use bisulfite, it's an all-enzyme workflow. So there are now methods, practical methods, for sequencing. And I'd like to emphasize sequencing genetics and epigenetics simultaneously on the same molecule. And the reason I'm emphasizing that is that one of the things I see in genomics is that people measure one thing, they measure another thing, and then another thing. And they overlap independent data sets and correlate it and draw conclusions. I'm not sure that's the best way of doing things. If you're trying to interrelate phenomena mechanistically, you have to measure them all at the same time on the same molecule.
Okay.
So you can do that now.
Yeah. Thank you.
Thank you very much. Maybe you can join me to thank Sir Shankar for wonderful work.